Um, Hi, everyone. Thank you so much for being here. This is the second session of our summer webinar series. For anybody who doesn't know me, my name is Anna Weber. I'm the Student Affairs Representative for the Society for Conservation Biology North America. So our webinar today is on handling genomic data in R. But before we get into that, I'm just going to give a brief rundown of what our organization is and what we do, in case anybody's unfamiliar. So this webinar is being hosted by the Society for Conservation Biology North America. We're a professional society dedicated to advancing the science and practice of conserving Earth's biological diversity. Biological diversity. Um, we envision a world with thriving biodiversity, ecosystems, and human connections. And our mission is to provide an environment that welcomes, supports, and empowers diverse students and professionals to contribute, advance, and apply conservation science. So what all that means, if you boil it down, is that we're a really broad organization with members from all kinds of backgrounds and all career fields related to conservation. We have biologists, we have social scientists, we have economists and conservation managers, government officials, really anyone who is involved in conservation in any capacity is welcome in our organization. Um, for our members, we host professional development opportunities like this one, like workshops, and we host conferences in North America every 2 years, with the next one being in Milwaukee in 2026. And that's in addition to the international conferences that the Global Society hosts. So SCBNA is a regional part of the Global Society for Conservation Biology, and they offer three different research publications that members can subscribe to keep up with conservation news and research. So if you're interested in learning more about society or about our society, or you think you may want to join, we have a QR code here that you can scan. It just goes to our website to give you more information and information about how to join if you're interested in that. So this webinar series has been organized, as I mentioned, by the Student Affairs Committee. Our role is to provide support and resources to students and early career professionals in conservation. Um, we do this by offering professional development activities like workshops and webinars like this one, technical skill building. We also have panel discussions with field experts, mentoring sessions again, for early career folks. And then especially at our in-person conferences, we have social and networking events. Um, so we have a really great group of people that work together on these programs. But we're always looking for new members. So if anyone's a member of SCBNA and you'd like to help out with sort of coordinating student-related activities, feel free to just shoot me an email. My contact's there at the bottom, and I'll also put it in the chat a little bit later. So, as I mentioned earlier, this is the second webinar of our summer series, which is focused on building technical skills that are generally available or generally useful to early career folks in conservation. So this one, as I said, is on working with genomic data in R. But after this, on June 11th, we have one on remote sensing data. And then from there on, they're pretty much every 2 weeks throughout the summer, with our last one, I believe, scheduled as of now in early August. Um, but the complete list of webinars we have upcoming is on our website. Um, yeah, there are more to come. We also have the links to past webinars that are on our websites. We had quite a few sessions on other topics of professional development, like grant writing and job applications, professional development or professional networking and career development. So feel free to check out our website or our YouTube channel. All the recordings of those webinars are there, and they're really great. So then before I introduce our speaker, one more thing I wanted to bring up is a project that we've been working on recently, and it's one that we could actually use some help with. Given the uncertainties around federal funding in the US for science and academia, a lot of early career folks are finding themselves without funding for their projects and with fewer opportunities for experience than there have been in the past. So we thought one way that maybe we could help out is to kind of crowdsource opportunities for students and postdocs, anyone early in their career who that are not reliant on US federal funding. So this is including graduate and postdoctoral fellowships or assistantships or scholarships, research opportunities for undergraduates, student and professional internships, and then small, medium, and large grants to support ongoing research. Um, so the goal of this is to have a list of opportunities that we can consider being relatively secure. Maybe they're funded through a state or through a nonprofit or a private foundation. And then anyone can add an opportunity to our list through Google Form, and that's what that upper QR code is for. And then if you would like to view the list, that's the lower QR code. And again, I'll put the links to these in the chat, and they are also on our website. We only just started doing this very recently, so there's not very much on the list yet. But the more people that contribute, the better it's going to get. So please, if you see an opportunity that you think is a good fit for this, please just go ahead and fill out the form, add it to our list. It's a very, very short form, so it would be great if anybody could help us out with that. And now I'm excited to introduce our speaker, Josh Janner. Josh is an evolutionary biologist who asks questions about the ecological factors that contribute to the origin, maintenance, and preservation of biodiversity. He uses population genetic sequencing, high-performance computing, and statistical modeling to address questions in conservation and evolution for a broad range of study systems, including bighorn sheep, butterflies, salgar, and woodcock. Josh is currently a postdoctoral research associate at the University of Wyoming and will be starting as an assistant professor at New Mexico Tech in the fall. Congratulations. Exciting. Um, so with that, I'm very excited to turn it over to Josh.
Awesome. Thank you so much. I'm so excited to be here. I'm also very nervous. So this is going to be really, really fun. I'm going to share a screen real quick and let's see if I can figure all that out. Rushing it. Maybe. Yep. Awesome. How does that look? Awesome. All right. So I just want to start off again by saying thanks for the invite to have me. I think this is a really cool series, and I'm so excited to hang out with you all today and talk a little bit about genetic data and how we might think about analyzing it. I want to start by giving you a fair warning. If my dog sees another dog walking outside, it will lose his mind. He'll lose his mind. He'll come in here and he'll start barking. It'll be the end of the world. There's nothing I can do about it. So, fair warning, there's pretty high chances of that happening. Um, so I just want to start off by giving you a quick outline of what we will be kind of doing today or thinking about. So I kind of have this broken up into four kind of main topics. The first one is just kind of talking and thinking about what the big questions in conservation genetics are and why we're kind of here today. Then briefly mentioning the most commonly used types of conservation genetic data, just kind of give you a quick background in that. Then I have two R scripts for us to kind of work through. One I'm going to walk you through, where we're going to simulate our own conservation genetic data, and then at the end for that last step, I'm just going to let you run free with a new R script, and we're going to explore a few different genetic analyses that we can use with our new shiny simulated data sets. And so yeah, when thinking about all this, I quickly realized when I was trying to plan what to do today that conservation genetics is just too big of a topic to kind of fit into I don't know just an hour and a half. And so I thought about how to kind of talk about those big questions in conservation genetics. And I really thought it would be more effective, instead of me telling you what the big questions are, to ask you what you think some of the biggest questions are in conservation genetics. And let's kind of brainstorm it together. So if you don't mind in the chat, please kind of I don't know, write out one kind of interest that you might have or you think is a really exciting question in conservation genetics. And then maybe also, since we're all becoming friends, let's maybe write down what your favorite animal is or favorite plant or something like that. And I'm very excited to see your answers. And I'm going to write in the chat too, if I can find the chat. And I'll just give us a couple more minutes while we think about it. And my dog's name is Harvey. So if I yell at Harvey, that's who I'm talking to. There are some amazing favorite animals right now. This is awesome. All right. Well, I think we'll keep going. I I'm really impressed with the big diversity of questions and interests that everyone has. I've found, so what I'm going to do next is I'm going to list out a few of them that I've come up with. Many of them you've already found or already listed here in the chat. But there are some that you all listed that I didn't even think of, which is amazing. The other thing is that this past spring, I participated in a conservation genetics reading group, and it was really fantastic. It was participants from many different universities, and we read a paper every week and really learned a whole lot about conservation genetics. And I found it really valuable. And so what I did is that on each slide moving forward, I've listed kind of a topic or a research question, and I've also linked a screenshot of a paper that we read in that reading group that I thought really fit well with that topic. And so now you'll have access to the slides moving forward. And if you really want to read more about a certain topic, now you'll have kind of a paper to kind of dig into. And so hopefully that will be a good resource. Yeah. All right. So here's a few of the ideas that I came up with. So the first one, well, let's see here, is genetic diversity and inbreeding. I think a few people mentioned that in the chat. I think this is one of the biggest questions in conservation genetics right now. In particular, thinking about how genetic diversity might relate to population declines or population growth or extinctions. I think this is going to be a major question in the field moving forward. Kind of along with that, we can think about identifying adaptive genetic variation, and that's variation that might be associated with local adaptation to the environment or some fitness effect that's really important for maintaining uh, adaptability moving forward in the face of climate change, which I think some folks mentioned as well. Another is just population structure and connectivity. Again, something that is really important, especially when thinking about how to manage populations in the future and which populations to manage and where those boundaries kind of exist. I think some folks mentioned population size estimation. So using genetic data to estimate the sizes of populations, I think this is really important for very rare species where it's really hard to sample individuals through time. And so this is something I don't know a whole lot about, but I think is really exciting. The next, and kind of going along with the population size uh, estimation, is demographic history reconstruction. So, thinking about way back in the past, when did different populations split off from one another? How have population sizes changed through time? This is really, really exciting work. I haven't done a whole lot of these analyses, but I would love to start thinking about this moving forward. Species delimitation is really, really important, especially given how a lot of the conservation framework works legally in many countries. So, for instance, in the United States, uh, for the Endangered Species Act, any population or species or subspecies has to be clearly delimited from other populations or species in order to be listed. And so, this is a really important kind of consideration when thinking about the legal frameworks of things. Hybridization is a potentially big threat to some rare species, especially when there might be an invasive species that comes in close proximity, or if there's another species that has a much bigger population. And so I think about this quite a bit in my research. And then one thing that I probably thought about the most is right now one of our best conservation tools is using gene flow or translocations or genetic rescue to move animals either back into habitats that they used to occupy or to increase and augment populations that aren't doing so well. And so, how can we use genetic data to choose which individuals to move and also which populations we might move individuals to and from? And then, kind of thinking from a long-term perspective, the more data we have through time, the more we can learn things because uh, oftentimes many rare species aren't at necessarily an equilibrium point. And so if we have data through time and we can use genetic monitoring, I think we can get a better picture of what's actually going on with our organisms of interest. And so those are just a few of the ideas that I kind of came up with on my list. But I hope this gives you just kind of a perspective, big picture perspective of some of the questions that are asked. Also, hopefully, you can see by some of the names of the studies that I put in these slides that conservation genetics is really applicable to a broad array of study organisms and research questions. And so I think it has tremendous potential to kind of make a positive influence in the world. And so it makes me feel really good about, I don't know, science and being excited to be a scientist, which sometimes, especially in today's world, isn't always the case. So yeah, it gives me a little bit of hope. All right. So the next thing I'd like to quickly go over is just kind of a brief, very short kind of history of conservation genetic data. And just so for those of you that might not be familiar with genetic data, just to give you a quick background of the different types and how it's changed so fast. So, probably some of the first genetic data that was used for conservation is just traditional DNA sequencing of mitochondrial DNA. And so, this was often used because there's a lot more mitochondrial DNA in a cell than for nuclear DNA. And so that makes it really easy to sequence. And all this is just you attach primers to certain parts of the genetic sequence, and you sequence in between those primers, and then you compare different individuals or different populations with one another in how they differ in the different nucleotides that you sequenced. And so this was a really common approach throughout the 90s and early 2000s and still used kind of widely today. But it had a few kind of drawbacks. First off, it was really hard to sequence more than, say, a sequence of a few hundred base pairs long. You can't get a long stretch of DNA really well with this approach. Also, mitochondrial DNA is only inherited from your mother in animals, and so it didn't quite give a clear picture of um, exactly how inheritance and ancestry was moving passed down through time, but also gave you some insights into maternal uh, dispersal and things like that. And so this was really great. It kind of laid the foundation for conservation genetic data, but we quickly moved on. And the next kind of data is kind of a really weird one. And so it's something called microsatellites. They are still very commonly used today. And they're also called simple sequence repeats or SSRs. And that's because what you're looking for or what you're sequencing is actually these repetitive regions through the genome that have a short kind of repeat. So here in my example, the repeat is just AGG over and over and over again. And the genetic analyses you do based on this type of data is all based on the number of repeats. So for individual one, it'd kind of be a heterozygote for 10 and 12 repeats, whereas individual two would just be a 14 repeat individual. And then you would do your genetic analyses based on that. So you're not even really using the DNA itself as the information for your analysis. It's just these kind of repeats. Microsatellites are somewhat difficult to set up because you need there's a lot of money that goes into it to set up the protocol for a given species or population. But after that, it can be really cost effective once you have it all set up to sequence a large number of individuals from the same place over time. And so it's because of that reason, it's still really commonly used. But since the advent of next generation sequencing data, this has slowly been replaced by those new studies. And all of that kind of really happened around 2008. The world really changed, at least in genetic sequencing. So here I'm showing you a plot with time on the x-axis from 2001 to 2022. And on the y-axis, I'm showing you essentially a cost of sequencing DNA on a large scale. So notice the large scale there. And what you can see is up from 2001 and even before then, up to about 2007, uh, the cost of DNA kind of decreased at a very steady rate on this large scale, and that was called Moore's Law. But then everything changed in 2008 when the first next generation sequencers came out. And everything became way cheaper to sequence. This was really, really exciting because for a long time, the next generation sequencing was only really kind of available for, human sequencing or kind of model systems in agriculture. And we couldn't really, use those same approaches for, say, conservation or wildlife populations. And this changed the whole game. And so now, uh, there's kind of been a revolution in conservation genetics to where we can easily access uh, a huge amount of data based on this new sequencing approaches. And so I'm just going to give you a quick kind of rundown of two kind of flavors real quick. So the first one is kind of a big, broad family of sequencing approaches called reduced representation sequencing. It's also called shotgun sequencing, or a few different other names. RADseq is one of the most commonly used approaches in this. And essentially, you're just subsampling different parts of the genome, and you're not getting a full genome approach. So what you do is you chop up the DNA in lots of different places using enzymes, and then you sequence right at those cut sites. And so at each cut site, you'll get sequences for an individual. And what you're looking for is places in the genome that vary at a single site in their nucleotide. And so the example I have here is some of the individuals have a T and some of the individuals have a G. And so this is what we call a single nucleotide polymorphism, or a SNP. And what we do is we sample throughout the entire genome at all these different places and get thousands or tens of thousands of single nucleotide polymorphisms that we can run our analyses on. And again, this is really effective because we're going to get way more data than we ever were able to get with mitochondrial DNA or microsatellites. Also, nowadays, it's a lot more cost effective to do this. The main disadvantage, though, is that much of this relies on bioinformatic processing. So there's a bigger learning gap to jump into these approaches. Now, you'll notice in the diagram on the right that I just put up that there's those question marks in between the cut sites. This just kind of is a reminder that we're only sampling certain places in the genome where those cut sites are, where those enzymes work. And so we're missing a lot of the genome variation, which can be a problem when we're looking for or trying to answer questions for particular genes or things like that. And so the next wave of sequencing approaches is whole genome resequencing. And this is actually uh, uh, much more expensive, but again, becoming cheaper and cheaper as every year passes. And essentially, we're sequencing DNA from across the reference genome. And I think this is kind of the future of where things are going. It's just going to take us a little bit to get there to where lots of labs are doing it. But some of the papers that I showed in the intro are kind of already doing these steps. And so that's kind of all the backgrounds that I want to kind of leave you with right now. And I think we should kind of start thinking about code a little bit. And I'm going to start thinking about how we can use R to simulate conservation genetic data that kind of looks like what you would get from a reduced representation sequencing approach. And so right now, I just want to kind of take a quick break. You can find the associated R scripts of what we're going to do at the link above. So please go ahead and kind of navigate to this spot and let's take just a quick break. If anyone has any questions about any of the topics that I've talked about so far, please put it in the chat and I'll see what I can do to answer that. Thanks.
Would you mind dropping that link in the chat?
Yeah. Let me try and do that. Everything in population genetics or conservation genetics kind of fundamentally boils down to this idea of allele frequencies. And so when thinking about that slide I had before, um, thinking about the reduced representation cut sites, right? And there were variable nucleotides at the spot. That is one locus in the genome. And the different nucleotides we have there are called alleles. And oftentimes in next generation sequencing data, we're only concerned with spots in the genome that have two alleles or called biallelic. And so I've kind of given you an example here of what if I went out with a butterfly net and tried real hard and I caught 10 butterflies. I created a sequencing data set, and I'm just looking at one site in the genome, right? And these are the genotypes of those individuals. So there's two alleles, either A or C, and the frequency of A in the population is just simply counting up all the different A's, which is 15, and dividing it by the total number of genotypes out there, which is 20. And so for this site, for this population, you would say you have an allele frequency of 0.75. And like I said, almost all population genetic analyses boil down to just allele frequencies, whether it be genetic diversity metrics or differentiation metrics or anything like that. It's really just this that we're kind of thinking about. And with our next generation sequencing data, it gets a little more complicated because instead of just thinking about a single spot, we're actually looking at thousands or tens of thousands of spots and thinking about the distribution of allele frequencies across the genome. And so this is what that might look like. And so this is an example from a paper that was published a couple of years ago by some folks that I work with. And this plot here is a histogram on the x-axis we have allele frequencies ranging from nearly zero to nearly 1. Um, Remember, we are only interested in spots with variation, so you can't ever have it exactly 0 or exactly 1. And then on the y-axis, we have just the number of loci in that bin with that type of allele frequency. And so this figure is called a site frequency spectrum or allele frequency spectrum. And this kind of is um, one of the hallmark kinds of metrics for population genetics. It's can be used to estimate demography of different populations. It can be used for phylogenetics. It can be used for, again, genetic diversity and for differentiation. And so if you have a distribution that looks like this, you can learn a lot about populations. And so, in an ideal world, if we wanted to simulate genetic data, we would want to simulate something that looks exactly like this plot here. And so, what we need is a distribution that we can fit to this plot to kind of give us a mathematical kind of summary of what this site frequency spectrum looks like. And fortunately, there's one really useful statistical distribution or probability distribution that we can use. You've probably heard about a lot of different probability distributions in the past from your statistics courses or other analyses that you run, like the normal distribution or the Poisson distribution. But today we're going to be talking about the beta distribution and why it's so useful in these cases. So the beta distribution is really great because it is a continuous distribution that runs from nearly 0 to nearly 1, just like our site frequency spectrum, right? And it can take a wide range of different shapes based on two parameters that you can assign. The first is called alpha, and the second is called beta. If you're following along in the R scripts, right now, these plots are all kind of based on starting at line 18. So if you're following along, we're kind of right in there. And in the R code, when I refer to alpha and beta, it's really, and the R code is called shape 1 and shape 2. So that's what it's referring to. So when alpha and beta are both 1, the beta distribution just takes the form of a uniform distribution. It's just flat across the board. Every value, every allele frequency is equally likely. But we can modify this to have all kinds of fun shapes. So if alpha and beta are exactly the same, but less than 1, then we have this fun U-shaped distribution, which kind of looks a little bit more similar to what we were looking at with our site frequency spectrum below. And the lower we push down alpha and beta, the lower down we push the probabilities of those intermediate frequencies. So there's more probability of being either really close to 0 or really close to 1, and less probability of being in the middle. We can also kind of skew these distributions to the left or the right by just changing the relative proportions of them. So by shifting either beta a little bit higher or shifting alpha a little bit higher. And all of a sudden, we're starting to get something very similar to what our site frequency spectrum is, which is really useful because if we have this probability distribution that matches, then we can use that to simulate our data. And so just as a quick kind of overview, this beta distribution is really useful because we can use it to kind of summarize data that is, kind of very similar to real life data. And so what I'm going to have you do right now in the code is just play around with line, I think it's 40. And what I'm hoping for you to do is to come up with alpha value and a beta value that kind of gives you a plot that looks like this. And so in that line of code, right now it says shape 1 equals NA and shape 2 equals NA. So try out a few different values of shape 1 and shape 2, and let's see what you come up with. And so when you're happy, when you think you're really close, go ahead and put it in the chat, and we'll see how close everyone can get to the true answer. And of course, if you have any questions at all, please let me know. Or if something doesn't seem to be working right, please let me know. And yeah, I can help out. Yeah, so there's a question by Jordan in the chat about using this to simulate data, or can we use this on observed data? Yeah, so you can definitely use it for both. And so um, yeah, and I use it for both in my research. I think it's a really interesting way to kind of summarize data by fitting a beta. I haven't included the actual code to fit a beta to empirical data in this code, but I could try to add that to the code and upload that to Git, and so that you would have it. But yeah, great question. Did anyone have any guesses? Ooh, there's first guess. Point 0.3 and point 0.09. Nice. Just give it maybe another minute, and then we'll keep going. Feel free to keep adding to the chat though. Oh yeah. Mhm. 0.45 and point 0.1. Yep. Oh, Harvey made an appearance. Here. Here. Here's Harvey. He is a Labradoodle. He's very needy. So, you know, he just got scared by the trash truck driving past the front yard. So, yeah. Um, all right. Oop, we have another one. Point 0.9, point 0.7. Awesome. Point 0.3, point 0.2. The trucks are very scary. All right, so I'm gonna go forward. So, like I said before, I fit this using another function that I should have added, but I didn't. So I'll add that to the script. Um, but what you get when you fit is a fit that looks something like that. So you're all really, really close. So based on this estimation, alpha is about 0.48 and beta is about 0.21. And so now that we have this beta distribution, we can create our matrix using that. And so what I'm going to do now is walk you through the function that you'll use to create your genetic matrix. And I believe that starts at about line 46 in the code. All right, so here's what that function should look like, what you should be seeing in the script. And I'm just going to kind of walk you through a couple of points. So the function is called single pop sim, and it requires you to include four different values. The first, in loci, is how many genetic markers do you want in your data set? Each row or each column in the data set will be one of those markers. The second one is in ends, the number of individuals. I think number of butterflies or bighorn sheep or whatever you study. Those will be the rows in your data set. And then alpha and beta are going to be those alpha and beta parameters, whatever you decide on in there. And so you could use the empirical ones or whatever you came up with to guess. It doesn't really matter. Okay. The next line of code is just making an empty matrix to put all the data in. Again, the number, each row is the number of individuals, and each column is a different locus. And then we just have a double for loop to make the data. And so the first one is for every locus, we are going to determine allele frequency based on the beta distribution. And so that is what's happening right there. And so we have our beta distribution that we plotted out before. And essentially, we're going to draw one at random from our beta distribution with a probability equal to that probability distribution that you plotted before. And so if you have one really, really skewed to the right, you might be more likely to draw an allele frequency of 0.9 than, say, of 0.1, right? Because that's what your distribution looks like. And so that's all we're doing. And then after that, for every individual using that distribution, we're going to draw from the binomial 2 times to determine a genotype. And so what the binomial does is it says essentially flip a coin and it'll either give you a 0 or a 1 back, right? And you give it a probability, which will be equal to our allele frequency we defined before. So if you add those up, it gives you a genotype. And so this is a little abstract and it's kind of hard for me to think about too. So let's go back to our butterfly example that I showed you before. So instead of thinking about our butterflies' alleles as A's and C's, I've instead put them as zeros and ones because the binomial, we're drawing zeros or we're drawing ones, right? And so if we reach into this jar of marbles to determine things, right, you can either draw a 0 or a 1. And the frequency of yellow and green marbles is based on our allele frequency that we drew from the beta distribution. And so if for an individual butterfly you draw out 2 yellows, then its genotype is going to add it together is going to be 0. If you draw out 2 green marbles, then your genotype is going to be 2, 1 plus 1. And if you draw one of each, your genotype is going to be a 1. And so we're just doing that for every individual and for every locus. So first, at every locus, we get that probability from the beta, and then we just draw twice out of the jar for every individual. And then we just do it a bunch, how many ever times we want that we specified in the function. And what you'll get is a matrix that looks like this. So again, each column is a different genetic marker, and each row is a different individual. And all this is just zeros, ones, and twos. And this may seem really, really simple, but this is actually the format that's required for almost every kind of genetic analysis. So if you want to try out different R packages or you want to create your own data or anything like that, this is what you would use to do that. And so you just have this kind of compact R function now that can make a matrix based on any kind of uh, site frequency spectrum that you're interested in, and for any number of individuals and any number of markers. And so hopefully this will be a resource that you can take and just kind of run wild with. And so that's kind of what I had for just walking you through that first script. And for the rest of our time, we're going to kind of I'm just going to let you kind of run loose with the second script. And the goals for that, the rest of our time, I think we have about 30 minutes or so, is to, in that script, you're going to simulate genetic data with the beta distribution for one or two populations. And you're just going to kind of explore the data for three different genetic analyses. I think I have calculating genetic diversity using expected heterozygosity. You'll run a PCA and create a PCA plot to look for genetic structure. And then you'll take one population, cross all the individuals, and see if you can recover parent-offspring relationships. And so, my hope for you is to kind of try out a lot of different combinations of beta distributions and number of markers and number of individuals and see when do you have the power to detect kind of real meaningful differences across populations. How many genetic markers do you need to get a good signal for different analyses and how that might change? And just kind of play around and kind of explore with how your decisions with your data or how the data looks might affect your outcomes. And so, good luck. I believe in you. I'll be right here. Feel free to either ask questions in the chat or hop on and ask a question. And yeah, I believe in you all.